All right, so uh, my title of my talk is misleading because it's about a renewed gut need for a genomic field guide to microbes. But really, what I want to talk about is birds, because birds are awesome and I love birds. Um, so I'm obsessed with birds, but in full disclosure, apparently I was obsessed about microbes even as a little kid. I found this essay in a pile of papers my mom gave me when she wanted me to collect all of my stuff. Isn't the handwriting awesome? This is from ninth grade English class where you can invent your own question. And I wrote, describe one step in the evolution of a bacterium as my question. So I guess I've been interested in microbes for a long time, but I've been interested in birds for longer. That's me not knowing how to use binoculars back when I was a kid. But I've been really into fieldwork and birds. And when I was in college, my first research experience was studying hummingbirds at the Rocky Mountain Biology Lab in Colorado. And I came back from that thinking I was going to work on birds. And then I discovered microbes again and started working on chemosynthetic symbioses involving deep sea organisms with Colleen Cavanaugh, and did research for a year after I graduated on that. And then I went to grad school and worked on microbes, and I've been working on microbes ever since. But I am obsessed about birds and still obsessed about birds. And I just have kept that interest going for a long time. And I just thought I would share. I've gotten into birds recently. I've started becoming a bird photographer, pretty amateur, I guess, but I'm obsessed about it. So there's a site called iNaturalist where I post my pictures. There's a group there for California birds, and the most observations of any birds in California are mine posted there, 27,000 something bird pictures, bird observations. So yes, I'm a little obsessed. Um, and I rediscovered my obsession actually directly due to my work on microbes. And it's not, we have been studying microbes on birds. I thought that would be a way to combine my interest in microbes and birds. And we've done a variety of studies on microbes and birds, but that's not actually what led to my rediscovery of my obsession about birds. And my rediscovery about that has come largely from work on thinking about field guides. So I'm going to tell you a little story about the bird microbe connection here related to field guides. So I assume many people here know what bird species this is. If you saw it out in, the field, you might say, oh, that's, exciting. Here's this bird. Maybe I'm going to try and identify it. People got excited one time when they saw this bird. And why is this? It was a little strange. It's strange to a birder if you don't know what this bird is that people would get excited about this. So if you want to know what kind of bird this is, you can look it up in a bird field guide. You can flip through your field guide. I have many of these out there. If you know where to look for that type of bird, you can find it. Here's the page with that bird. It's an American robin. The field guide shows you where they're found, gives you all sorts of information about the bird. If you're a dork like me, you can have a bird app on your phone. Or this I took actually 2 days ago outside my house and posted the picture to iNaturalist. And iNaturalist helps you identify the bird by looking up by the picture with an artificial intelligence system, what similar things might be in your neighborhood. And then the community of people out there help you identify the bird. Here, someone else helped identify it as an American robin. All exciting, right? It's an American robin. It's boring to most birders because it's really common and really abundant. And who cares about the common and about, actually I do, but most birders don't. So let's go back here. Why were these people excited about seeing a friggin' American robin? Well, if you look at this picture carefully and you're really insightful, you might notice the cars are parked on the wrong side of the road here. This is in the United Kingdom in 2004. And the people there were incredibly excited because that is, after all, an American robin. And if you look in the Birds of Britain bird guide, the American robin isn't there. It's not supposed to be there. It's out of place. So they were incredibly excited because, they have friends who told friends and they said, look, there's this rare, exotic bird showing up out here. Isn't it great? We should all come look at this bird. And get excited about it because many have never seen this bird before, even though in the United States, it's just like the garbage bird, the boring bird. As an aside, I didn't tell you there's another bird up here. That's a falcon. And amazingly, all these birders went crazy, and then they proceeded to watch the falcon eat the American robin right in front of them. So the bird was out of place, but didn't do particularly well in its exotic location. But that's not what I'm here to tell you about. I want to talk about the implications of this, the excitement of finding, a new species. And people do this all the time with birds in particular, but with other organisms. So you can subscribe to alerts. There are WhatsApp groups where you can get notified within minutes of a new weird bird showing up in your neighborhood. There are Google groups, email groups, Twitter handles, and all sorts of things where you can find out the, rarities or something being out of place in some sense. And you can do this with other organisms too, right? There are field guides to all sorts of organisms. I also collect field guides, so I'm obsessed not just about birds, but you can do this with lots of other organisms. And the strength here is this is made possible by field guides. And what I want is a field guide to the microbes, a real field guide to the microbes. And what I'm going to walk you through is what this would include and why I think we can do it and how we can do it. So what is generally included in a field guide? It's a catalog of organisms, right? For whatever particular group, let's say birds, it's a phylogenetic catalog. So they usually list them by their families or something to that effect. And it's a list of all the organisms in a particular place or most of the common organisms in a particular place. It has information about their functional diversity. That is what behaviors they have, what they eat, what they do, how they fly. It has information about their biogeography, like the map I showed you with the American robin in both space and time, where you can find them. It has information about sort of the micro niche. That is, it's not just found in California, but robins, are not found swimming around in the water. They're found in trees or something to that effect. And it has the means for their identification. So a guide to how to identify these organisms. And this is really important for many reasons, but what I'm going to walk you through in particular is it allows you to interpret the normal states for those organisms and also understand abnormalities. So for microbes, that would include things like many of the talks today and posters and focus on emerging pathogens. How do we know something is emerging? We know what it used to look like and what it looks like now. It's also really important for finding new functions. If you know where organisms are found and where they prefer to be found, you can target your discovery that way. Critical for forensic applications, for studies of climate change, for understanding the spread of antimicrobial resistance. One that I won't talk about that I used to work on quite a bit was how you can try to determine if an organism has been genetically manipulated. You need to know what's normal in order to identify what's abnormal in an organism. And for detecting things like bioweapons release, you need to know that anthrax doesn't belong in the air in the subway system. And you can do things like vaccine design. So I got to pause for a second here. Just a family thing. Hang on one sec. You still there, everyone?
We're here.
Yeah, sorry. Little knee skinning emergency here. Sorry about that.
Oh no.
Life is real.
It is real. Do you need to go?
Right. No, it's fine. It's
okay.
Fine. Sorry.
Yeah.
So anyway, as I was saying, just going back a second here, I think that for many reasons, we actually want to do this field guide to the microbes like I was walking through here. And so I thought this was like 20 years ago, we should just do it. We should just do this field guide to the microbes. And then I thought about it some more, and it's deranged. It is completely insane. So like, I don't know if you can see over my shoulder, I have like field guide to the birds. In the United States, there are some 800 or so bird species that you typically see. In the human gut, there are probably 20,000 bacterial species, another 50,000 viral species, all sorts of microbial eukaryotes, yeast, and other things. In one single sample, there's more species than there are birds on the planet, basically. And of course, there's incredible diversity. So, you know, is this crazy? I wrote papers about this. And then as I was writing the papers about this, I realized it's crazy. And, you know, one of the challenges is, of course, they're small. They're not like, you know, birds, you know, hummingbirds are small to birders, but they're much bigger, of course, than microbes. There's incredible diversity of form across the microbial world. There's an incredible diversity of functions. So if we want to catalog all the functions as part of the field guide, it's quite more diverse than what you see in birds. And there's evolutionary diversity. There are, you know, the cellular organisms all across the tree of life include microbes from every branch. And there are, of course, the viruses, as many of the people at this meeting are working on. There's just an insane amount of diversity. I mean, there are some estimates that there are, you know, hundreds of millions of species of bacteria. So, you know, obviously a field guide to the microbes isn't going to actually include all of these organisms. There's also, you know, localization issues. So it's not like you can say this particular microbe is found, you know, on the human skin, and that's all you need to know. You need to know subcellular, sub-regional localization to really understand the diversity. So it's crazy, right? And of course, most of what we know about microbes is when we rear them in the laboratory by culturing or some other method. And, you know, it's not always possible to do that. There are alternatives like DNA sequencing that I'll talk about in a minute, but there's just a lot of challenges with looking at microbes. So again, building a field guide to microbes is an insane, insane idea. But then I thought about it some more and I realized it's a great goal. Even if we can't achieve it, it's a great organizing theme. And it is, in fact, possible to start to build some of the components of this. And what I want to walk you through are some scientific and technological solutions that have come up in the last 10 or so years that continue to make this not as crazy as it seemed, and some solutions related to community engagement. So in terms of the science, the thing that I focus on in particular is DNA sequencing, and as everybody knows, DNA sequencing keeps getting cheaper and cheaper and cheaper. This was, you know, particularly catalyzed by the so-called next generation of sequencing that started in about 2006. And it still continues to accelerate, maybe not as faster than Moore's law as it was doing for a while, but DNA sequencing is a powerful tool for characterizing microbes, and it's just gotten cheaper and easier, including to the point that, you know, there's now a thumb drive DNA sequencer from Oxford Nanopores that you can go out and use in the field. So we've started doing projects that I would consider one component of a field guide, which is to generate genome data from across the phylogenetic diversity of microbes to fill in the branches of the tree of life that don't have genomes. First did this before I moved to UC Davis at a place called Tiger, the Institute for Genomic Research. When I moved to Davis, I have an adjunct appointment at the Joint Genome Institute, and we started a project called GEBA, the Genomic Encyclopedia of Bacteria and Archaea, to really fill in genomes from bacteria and archaea for cultured representatives from across the diversity. There's now dozens of spin-off GEBA projects. They're still doing this at the JGI. So we're filling in the tree for bacteria and archaea for cultured organisms. And of course, there are many other projects. I'm not the only person doing this. There are thousands of projects where people are going out and surveying diversity. Here's a recent example with SARS-CoV-2 and relatives from the UC Davis and related group PREDICT project. But lots of people are doing this, going out and trying to catalog diversity, partly through DNA sequencing. So with DNA sequencing, we're starting to fill in the phylogenetic catalog. You can predict functional diversity, although you don't know it. You get information about the means for identification from this too. As people go out and do environmental sequencing, by grinding up samples and just without culturing, directly sequencing from the environment, we start to get the biogeography information for microbes, that is space and time. And as we apply other technologies that have also advanced, like microscopy and high throughput culturing and microfluidics and environmental surveys and other things, we're starting to fill in the studies of the niche and really fill in some of these things that are required for a microbial field guide. Still imperfect. And obviously, again, you're not going to do this for 100 million species at a time, but people are really starting to progress on these components that you could actually use to start to understand what is normal, what is the background, and then start to understand what is different. And when you ask what is different, it's not just about an organism showing up in the wrong place. It could be the wrong genetics. That's for like genetically engineered organisms or hybrid species, as we see for plants and animals, and so on. But what I also want to talk about is the need for engagement. So with the birding community, one of the reasons I rediscovered birding was because the birding community is so good at engaging the community in part of the studies of biodiversity. And we've been very interested in my lab for the last 10 years in figuring out ways to carry out citizen microbiology projects that are mimicking, at least in part, what goes on in the birds and butterflies and gardening and plant biodiversity and such. So we've done a few of these. We had a kitty microbiome project run in my lab by Holly Gans that actually spun off into a company that she now runs called Animal Biome. We've done a seagrass microbiome project. We've collaborated on projects to survey phones and shoes across a bunch of events, collaborating with a group called Science Cheerleaders that go out to sporting events and educate people about microbial diversity and then have them collect samples from the environment and so on. There are probably 50 at least other large-scale citizen microbiology projects out there, maybe hundreds now. Lots of people are starting to try and figure out ways to engage the broader community in studying microbes and in collecting samples. So crowdsourcing is an important part of this. And in getting people to think about microbial diversity in similar ways that they might have been thinking about bird and butterfly and plant diversity. It's still challenging because, of course, you can't, in many cases, for most of the microbes, you can't really see them in any useful way, even if you have a portable microscope or something like that. And identification of them through, say, DNA typing is not something everybody can do by themselves in the field, but you can engage people in looking at the data once it comes back and in thinking about the results. And this is a growing area of microbiology that I think will contribute to the creation of a field guide. We've also thought about many other approaches to engagement. I don't know if you can see over my shoulder, David Coyle in my lab made a microbiome board game. It's not as amazing as the new bird board game that everybody is playing, but it's still, we like it. Still think it's really cool. We think there's a much bigger need for engagement of the public and thinking about microbial diversity. The pathogens many people think about, especially right now, but we want them to think about all the other organisms too. So again, are we at the point? I actually think we're getting to the point where we can think about having all of these things for at least some groups of microbes in some locations and start to build this catalog that you could call a field guide to the microbes. So I really do think, it's still crazy, but I do think that this is possible. So I like the boring organisms. All these birders who chase after the rare organisms, I don't do that. I like to just see the same types of organisms over and over. I go out to YOLO Bypass near UC Davis like 4 times a week. And I have 90 observations of American robins that many of my birder friends are like, why on earth would you do that? Why would you observe the common organisms over and over and over again? Because we have to know what their diversity is. We have to know where to find them, and we have to know when they're anomalous. So I'm hoping that people start to think that not all microbes are boring either. I collected pictures of some of the microbes that people were talking about today. Stole them from the internet. I apologize if I stole one of your pictures. But microbes are not boring. We need people to catalog their diversity, their genetics, their evolution, their function, their niches, and we can do this. We can build a field guide to the microbes. And I will end there. There's my lab. Thanks to all the people in my lab and all my collaborators and funders.
Thank you so much, Dr. Eisen. That was a really interesting talk that I think got a lot of us just thinking about so many questions to ask you. I wanted to see if everybody could put their questions into the chat. We already have, do you have time to stay with us for a few more minutes?
I do, yeah.
Okay, sure. Great. Marta Schockett put in a question that almost much more eloquently than I would have said gets to my question about this issue of species names, because the field guides, of course, for the birds, it's pretty well defined what's a different bird species, and I think we do have the subspecies context for animals, but when it comes to microbes, that is a very frustrating issue for many of us who try to work on these things. So I think, so Marta's question, I think you can see, is first of all, when there's new discovery and it's largely based on genome sequencing, maybe sometimes a part of the genome, how do you handle species namings? And what do you think is the sort of best approach there?
Yeah, so I mean, maybe I can post on in the chat later. I'm part of a group that Anna Louise Reichenbach and a few other people organized to work on how you name microbes with genomic data and without cultured organisms. We published a review paper about this a few months ago. There have been a few other review papers about this. So it is challenging. And the way you name bacterial species, you have to follow a set list of guidelines that don't really work for genomic data and sequence data. So there's a bit of a controversy over this and some fights going on. I think everything should be named with a phylogenetic context. I mean, obviously that is how most organisms are named in other groups. Where you draw the line between species is challenging, but feasible actually for microbes. Generally, you see, like you do with multicellular organisms, genetic boundaries that are pretty clear between species that recombine within but not between groups. And you get hybrid zones, and you get, lateral gene transfer makes things more complicated, but it is feasible to do this. I think that it is also important to point out that this is a continuing struggle in plants and animals. So just for the record, I did a prank many years ago related to a bird group called the Dendroica warblers that a friend of mine studied, and I created a fake named person whose last name was Dendroica that sent him some emails about this. The Dendroica warblers don't even exist anymore. They have been renamed to some other genus. They've been deleted from all the records. So like, if I tried to pull off that prank now, it wouldn't even work because that name is gone. It's been erased from the list of all birds. And people are renaming, like bird species. It's not, as challenging as with microbes, but it is an issue with plants and animals. But it's a really good point. And I will post links to some of the papers that people have been working on to try and deal with this.
Yeah, that's great. It's so good to know that you're in there working to address it. I guess in your perspective, how different is the scale of the problem when you go from bacteria to viruses? Like you've done some work on viruses. It's just, to us, it becomes even exceedingly more difficult to think about.
Yeah, I mean, the whole challenge, I mean, viruses are another complication because first of all, it's not easy to place them all onto a single tree. So the phylogenetic classification is challenging across the diversity, but you can do it within groups, within, subgroups of viruses. You can do this, so like SARS-CoV-2, you can do the alpha, beta, gamma, delta coronaviruses, and then zoom in on the betas and do things like that. So some of it is similar. The scale of numbers of cells, right? A number of virions is enormous. The total genetic diversity is great, but it's still, I mean, I think the PREDICT project and the other people that have been surveying coronaviruses show a good example of why you want to know what the natural diversity is and why you want to know where it's found and why you want to know some functional information about it, like all the predictions about spike protein attachments and things like that. So the goal is similar, like to try and organize it around a field guide, I think is a really useful goal. And of course, viruses have the same issue if you see the arguments about when we should name a new variant and how we should name the variants. It's a similar challenge. So I think it's probably harder for viruses globally, but you can pick a single group that, like with birds, right? I'm not talking about a field guide to all microbes at once is, I think, an important point. You have to narrow it down, like either do the cyanobacteria in all lakes or something that narrows it down rather than trying to tackle everything at once. So you could pick coronaviruses in animals or leishmaniasis, like we've already heard talks about trying to find the host range of various eukaryotic parasites and define that. You could just pick that as your target and build up the field guide for them.
That makes perfect sense. Let's see, we have a couple more questions. I think we have time. So Chloe Rickards asks, have you reached out to Foldscope? They make $1 microscopes and they have a growing online community.
Yeah, so because I had too many slides, I deleted the last slide I deleted was about Foldscope. And they're amazing. Manu Prakash and the group behind Foldscope is incredible. They are one of the best examples of participatory science where people are looking in part at microbes. Most of the people using Foldscope aren't looking at microbes, but there's a large collection of them that are. Diatoms and fungi and ciliates and a variety of other things. And that is an incredible example of an engaged community and a tool that enables people to start to identify some things. I mean, the key challenge is that microscopy-based identification only works for certain types of microbes. But there are communities that have been built up around the Foldscope that are focusing on those, and that's like the poster child. I shouldn't have taken out that slide, but they are amazing.
That's okay. You got it in now. Really great to see how the citizen science is being channeled for good all over the place. One more question. I think we have time for just that. So Erin Mordecai is asking, beyond the microbial genomes, are methods advancing in terms of identifying microbial ecological niches, functional traits, geographic distributions, behavior, and other factors normally found in the field guide? Oh, that's such a good question.
Yeah, I mean, I think that this is, I sort of skimmed over this, but this is, I do think that people are starting to do this with things like functional microscopy, where people are actually trying to look within individual niches for where particular microbes are and what those microbes are even doing. So like nanosims technology and some of these other microscopy methods where you can track the movement of isotopes within microbial biofilms and things like that. There's some really great work where people are, going from information you have from genomes and or cultured organisms to really trying to understand what's going on in the real world in terms of the functional properties. And, some of this, you can do it, indirectly by exposing an environmental sample to, certain compounds and just seeing what happens to them and seeing if your models are correct. But I really like the ones where they're trying to subdivide this up with sort of enhanced microscopy methods for doing that. I think that we're getting, more data about culture diversity of microbes that will help inform some of that, because one of the biggest challenges for a while was we generated all these genomes and then you try to predict the function of organisms, and your functional predictions are based upon studies in E. coli and B. subtilis and some other organisms, but not a wide diversity of organisms. So we need much more genetics and biochemistry and functional studies of across the diversity of life in order to then make predictions for environmental studied organisms.
Excellent. Just really, really enlightening discussion. Thank you. We're so happy to have you as part of the Bay Area EID now, hopefully, for long term. And thank you so much for that.
SARS-CoV-2 has made me rediscover my love of ecology and evolution of infectious diseases.
Perfect. I think, yeah, that it's becoming very widespread. So that's wonderful.